Welcome to today's lecture on Sanger sequencing in molecular biology. Sanger sequencing is a widely used DNA sequencing technique that allows us to determine the order of nucleotides in a DNA molecule. In this comprehensive lecture, we will discuss the various components and steps involved in Sanger sequencing, including dideoxynucleotide triphosphates, ddNTPs, DNA polymerase, fluorescent dye-labeled terminators, electrophoresis, automated DNA sequencers, troubleshooting, quality control, big dye terminator chemistry, data analysis, advanced applications, comparative analysis, and future directions. So let's dive in and explore the fascinating world of Sanger sequencing. In Sanger sequencing, ddNTPs are used as chain terminators to halt DNA synthesis. These modified nucleotides lack a 3O group, inhibiting further extension of the DNA strand. Each ddNTP is labeled with a unique fluorescent dye, allowing for identification during electrophoresis. DNA polymerase plays a crucial role in Sanger sequencing as it synthesizes the complementary DNA strand. A thermostable DNA polymerase, such as Taq polymerase, is commonly used due to its resistance to high temperatures required for denaturation. Fluorescent dye-labeled terminators are modified versions of nucleotides that terminate DNA synthesis. These terminators are covalently attached to ddNTPs, allowing for accurate identification of the incorporated base during sequencing. Electrophoresis is a key step in Sanger sequencing, where DNA fragments are separated based on their size and charge. By applying an electric field, DNA fragments migrate through a polyacrylamide gel or capillary tube, with smaller fragments traveling faster. Automated DNA sequencers have revolutionized Sanger sequencing by streamlining the process and increasing throughput. These instruments incorporate capillary electrophoresis, laser excitation, and fluorescence detection, allowing for precise and automated sequencing. Sanger sequencing experiments may encounter various challenges, such as poor sequence quality or ambiguous results. Troubleshooting techniques include optimizing primer design, DNA template preparation, polymerase choice, and reaction conditions. Maintaining high-quality sequencing data is essential for accurate results. Quality control measures involve assessing DNA purity, quantification, template integrity, and the efficiency of sequencing reactions. Big dye terminator chemistry is a widely used method for DNA sequencing. It employs modified dideoxynucleotides labeled with different fluorescent dyes, enabling robust and accurate sequencing of DNA fragments. Data analysis of Sanger sequencing involves base calling, alignment, and assembly of DNA fragments. Specialized software, such as Sequencher and Nestar, are utilized for accurate interpretation of sequencing data. Sanger sequencing finds diverse applications in genomics and molecular diagnostics. It enables gene identification, detection of genetic variations, characterization of disease-associated mutations, and studying DNA-protein interactions. Comparative analysis of Sanger sequencing data involves comparing sequences from different individuals or species to identify genetic differences or similarities. This approach aids in understanding evolutionary relationships and disease susceptibility. The future of Sanger sequencing lies in developing higher throughput methods, reducing costs, and improving accuracy. Advances in nanopore sequencing and single-molecule sequencing techniques show promise in overcoming current limitations. Indispensable tool in molecular biology, enabling to explore the various components and steps involved in Sanger sequencing advancements. As research progresses, and contributing to advancements in genomics and molecular